Hello, in this video we're going to be talking about prokaryotic transcription. Let's not waste any of your time. Let's get right to it. Now, when we're talking about prokaryotic transcription, one of the things that we're going to focus on in this video is the ways in which they are similar, and that's a long list because it's mostly similar, but we're also going to be looking at some of the ways in which there are difference. There's a difference between the two, just to highlight those. But initially, as one of the similarities to eukaryotic transcription, there are three major stages: initiation, elongation, termination. And we know the first two are fairly short, quick processes, and we know the middle one is the long, tedious part where all of the work is being done. Now, during initiation, if you remember that eukaryotic cells had transcription factors, okay, the same type of thing is going on here. The enzymes alpha prime, B prime, those are associated with getting things started, initiation, and of course you've got an omega enzyme, which is more about the transcription process kind of during elongation, but it attaches at this time. So you've got your polymerase that binds to the promoter, get it started, again just like eukaryotic transcription, and of course that gets the thing started. But again, a lot of similarities there, different names as far as what they may be, but like the process can be the same. Who does that is maybe a little different. Okay, but during initiation, and again that diagram is maybe kind of small, but it's also in your PowerPoint if you want to look at it there. But you can see, and maybe I can point this out in an easier fashion. Obviously your first ribosome on the far left, that's the when we saw on the last screen, that's when everything is kind of adding to it. So now your middle one, you're looking at the -10, the -35 sequence, which is kind of in that yellow highlighted section there. It's kind of like that's the start, and then of course the -10, that region between those two, that's kind of the starting point, the promoter, and of course that's called a consensus sequence because it has the same function in all bacteria, but that word consensus sequence isn't on the diagram. But just like eukaryotic cells, the DNA is unwound, the mRNA, and it's red, transcribed into the RNA material, and then of course it's wound back up. The DNA strand is as well. Now, each of those subunits that we talked about, the alphas, the omegas, the betas, they're all very important. And if they're not present, then jobs can't be done, and then something is wrong, with the mutation has occurred or whatever, and now just simply the cell will no longer reproduce RNA strands, and of course it will terminate. Now you may think that, boy, that sounds kind of harsh. If one of our cells didn't work, yeah, it would just shut down. Of course, the difference is we have, trillions of cells. Well, guess how many cells the bacteria has? Just the one. So it's pretty detrimental to the organism at that point in time. But again, another similarity, just like eukaryotic cells, that TA box, it's part of the initiation process and to kind of get that thing started. Okay, stage 2, elongation. Okay, again, just the same name, same situation going on as with the eukaryotic cells, and you can see here again the DNA is unwound, and you've got the RNA, the DNA being transcribed into the RNA. You've got that transcription bubble again, red just like in eukaryotic cells. Polymerase is doing the job, the DNA template, and in the RNA strand that's being grown. So you can see a lot of similarities between the two. Maybe not the worker bee is always the same, but the same process is going on. All right, the idea of termination. Checking my slide here. Yeah, I think I almost got off there. The idea of termination. Now here visually you can see some different, or a major difference. You can see what the RNA strand is doing there in the diagram. Of course, the termination begins when you reach that stop codon, just like with eukaryotic cells. DNA rewinds, polymerase jumps off the RNA strand, but then you can see the RNA strand in prokaryotic cells. It kind of folds onto itself. Okay, eukaryotic cells, this doesn't happen, and what allows that is there are the UA base pairs towards the end are weaker, so you can kind of fold that back around, kind of make that little hairpin. But the idea of that being a major difference. Okay, another major difference between the two is that transcription and translation, they can't start at the same time, obviously, but they're going at the same time. So as the polymerases are going down the RNA strand, making the RNA strand, ribosomes come in and attached to that RNA strand, and then begin to build their amino acid chain. So that's a pretty efficient system to when you have, once the RNA strand gets long enough, now the ribosomes start to read it. And you can see that in the diagram beside me, that far right hand side, you can see that. You can see the polymerase, that's the green circles, and then what's hanging down below, that yellowish, orangish stuff, that's the RNA strand being read. And then the one on the far right, you can see purple strand coming off of that. That's the amino acid chain being done. So it happens kind of in a simultaneous process, and of course that's different than us. Ours happens not only separately, but in different locations as well. The transcription happens in the nucleus, and the translation happens out in the cytoplasm. And you can see there that the RNA-based enzymes, it's the ones that kind of catalyze or get that translation started. Well, that's our video over prokaryotic transcription. It's also our final video in this video lecture series. When we come back next time, we're going to be talking about plant structure. We'll see you then.